Welcome to the lecture series in food biotechnology. In the last lecture, we saw the different breakthroughs of the biotechnological applications, how hybrid corn was discovered, the term gene cloning, what it meant with, then the application of artificial insemination, to name a few. Then we had the discovery that microbes can be utilized to produce drugs. You had the first recombinant vaccine, Humulin, the human insulin, which was a recombinant product, commercialized. So, all these discoveries came in the later part of the 1990s. Let's see now some more applications of biotechnology. In 1990, we had the first federally approved gene therapy treatment. This was done on a young girl for an immune disorders. So, immune disorders are where your own immune system fights against you and kills your self cells. And this sometimes happens by spontaneous mutation, or it can be congenital, what you inherit. So, you remove the bad gene and insert the good gene. So, wherever the damage has happened, you remove that and then insert the new one without any damage, the patient will get cured. Scientifically, this was proved, this was accepted. The knowledge that mutations can be inserted further gave a proof to this, and this subsequently led to the development of gene therapy, which was approved and done for treatment of this young girl. And in 1994, the first transgenic or genetically modified food crop was approved by the US Food and Drug Administration. This was the flavor saver tomato. This was the name of the tomato crop which was approved. Now, as you know, tomato gets rotten or spoiled very easily, and this is because fundamentally the fruit has a very thin outer coat which can get damaged due to bad handling, it can get spoiled, and normally the fruit sustains only for a couple of days before it gets rotten, before it gets damaged or spoiled. So, when you are trying to transport from fields to places which your consumer places for, you have a huge loss to the crop because of spoilage during transportation. So, the major thing now was to prevent, how to prevent this fruits getting spoiled, and scientists found that this was because the outer coat, you had a pectinase which actually acted on breaking down the pectin in the outer coat, which further deteriorated the quality. So, here they silenced the gene in this genetically modified crop, they switched off or silenced the gene for this pectinase, so that the detrimental development or deterioration of the quality of the fruit will not happen. So, what happened was, your ordinary tomato, normal tomato, within 10 days, in 10 days this is the status. In 20 days your ordinary tomato started, the outer coat started getting shriveled, the inner portion getting damaged. In 45 days it becomes completely spoiled. But the genetically modified or the gene silenced tomato, even after 45 days it was intact, which meant that improved shelf life. The loss due to transportation spoilage, the deterioration of the quality could be prevented because of that. So, this the first approved genetic crop. Here what they did was, they introduced what we call as an antisense RNA. So, you have, in biotechnology terms, you have a gene producing an enzyme in any organism. So, this we state this enzyme is formed because of the sense strand. Now, if you introduce an antisense strand also, the sense and the antisense will bind together, which will get destroyed. So, the product will not be formed. This was the technique which was employed in the production of flavor saver tomato, and this was commercially approved in 1994. And in 1997, scientists of the Roslin Institute, led by Ian Wilmot, cloned Dolly, a sheep, from 2 adult sheep cells. That a live organism can be cloned, and it was a fully functional organism. This in 1997 was a major breakthrough. Then subsequently in 2001, you had the Human Genome Project and the Cellular Genomics created a draft of the human genome sequence. This was published. So, all the 46 chromosomes of the human genome, their sequence was published both in Science and Nature. And among plants in 2002, rice was the first crop to have its genome sequenced or decoded. In 2003, the Human Genome Project was completed, providing information on all the locations and the sequence of human genes in all the 46 chromosomes. And in 2008, the Japanese astronomers launched the first medical experiment module called as Kibo, which was used in the International Space Station. So, this is a development on the other side, where the first medical experimental module was developed and launched. Now, if we take up the recent breakthroughs, the development of a biosensor is very exciting. This biosensor utilizes microbes or monitors or electrical pulses which can be converted to track various biosignatures like blood pressure, pulse, breathing, body temperature. So, this term, the development of the biosensor is not only important in terms of medical field, but it has potential applications in all aspects, including agriculture, bioremediation, in all the aspects we talk about. And an innovation company called as Sensible Medical have developed a vest monitor. So, especially in people with failed heart who are chronic, who are vain, you put on this vest. So, this vest has a biosensor. So, this vest will monitor lifetime the amount of fluid in the lungs, and then whenever it reaches a critical level, it will immediately alert the doctors so that the patient can be saved. So, instead of manual monitoring, this becomes a live monitoring, which becomes very easy. So, the development of the biosensor is a huge breakthrough which can be utilized in many fields. The other aspect we were talking about is 3D bioprinting. Now, this also has potentially huge potential to create drugs, to pills, artificial organs. So, here what we do is we use the living cells, and using the technology of 3D bioprinting, we can create human body parts like the heart, heart valves, skin, cartilage, which can be used. So, organ transplantation now becomes easy because we do not have to wait for a match. We do not have to go for rejection. You take your own body part, use 3D bioprinting to create part of whatever plant you want to replace and utilize that. Since the living cells are from you, there is no chance of rejection. Bioprinting also has the potential to create drugs and pills, like entire organs like hearts or life grow from the patient's own body cells. So, the ability to print human organs will also have applications in medical research and training, and as a way to provide accurate samples for study and analysis. This is a human bioprinted ear which has been grown. So, like that you have huge potentials to this particular field. The other aspect we are talking about is bioplastic. Environmental and ecological concerns are fast becoming a major area of focus. As you all know, ordinary plastic takes hundreds of years to break down. It is a huge threat to marine life. So, we are replacing the ordinary plastic with biodegradable plastic, which gets degraded easily. The source is also easy for us. So, bioplastics, where we have utilized plant resources to make the bioplastics, we produce durable products. Along with this is your edible cutlery. Instead of disposable plastic cutlery, we have edible cutlery getting developed. We can eat your plate and spoon utilizing this. That is also the second aspect. The next aspect would be biofuels, bioenergy. So instead of depending upon petroleum, the fossil fuels which are fast depleting, we try to create bioenergy, biofuels. So here what we try to do is, we produce, in sustainability aspect, we produce biomass for the production of biofuels and bioproducts with minimal to positive impact on the environment. For this, the next biomass development, we develop crops with cell walls optimized for deconstruction and conversion to biofuels and bioproducts. So we develop the plant biomass in such a way that they can be, after their crop production or yield is over, they can be easily used for converting into biofuels or bioproducts. Then the breakdown of this, improve the microbes, improve the enzymes in such a way that this biomass developed can be easily broken down. Then the biomass can be converted into sugars and lignin, which can be easily recycled back. So here we are trying to improve the enzymes and the microbes which degrade this biomass. The further aspect would be biofuel and bioproduct synthesis. So here we engineer the bacteria. We manipulate the metabolic pathways in such a way so as to produce more amount of biofuels and bioproducts. All this is also part of utilizing biotechnological applications. The next aspect is gene editing. This is a term. The CRISPR-Cas9 technology is something which even layman would have heard of. The CRISPR Therapeutics hopes to utilize genetic engineering to help millions of people afflicted with hereditary diseases. This platform is called as CRISPR-Cas9. This is used to cure ailments through gene therapy. And in addition, the CRISPR Therapeutics is planning to offer their applications of this platform in utero and in vitro patients, not only to screen for hereditary diseases before birth, but also try to find a cure for them in treatment. The other major breakthrough is virtual and augmented reality using biotech innovations. These systems work across medical fields, for example, by assisting surgeons during delicate operations or for helping patients recovering from injury. The virtual and augmented reality allow people to examine the 3D representations of the human body and its various parts. So when you can see the representation, we have a more effective demonstration of medicine and its effects, and this lends a greater assistance to doctors during diagnosis. That is, you use this virtual and augmented reality, simulate that you are giving a drug, then trace out the pathway of the drug through the human system, then find out what it is doing inside. So this will give you a better way. Suppose your drug has a huge or lethal effect. Even before trying it out on patients live, your simulation using virtual and augmented reality will tell you that these are the drawbacks of the drug. So now, without wasting time, you can go back to the blackboard, find out how this defect can be rectified, do that, and then bring it back to the commercial aspect. In virtual reality, you can experiment with dangerous bacteria or combustible materials with no risk, because it's only in virtual. You are not experimenting it live. And you can also forget about any worries about breaking anything or harming anybody, because you are using a very dangerous pathogen or a combustible material or a potential lethal pathogen. So here everything is done in the virtual field, so it's safe. This technology also allows the scientists to broaden their understanding of previously inadequately studied biological interactions on a microscopic level via detailed 3D models and virtual reality headsets. So whatever you want to work out, you can work out using virtual reality, find out whether it works, what are the drawbacks, then try to improve on the drawbacks, then go back to the reality, virtual reality experiment. So the product ultimately which comes out would be safe for human use. So this is a huge, this has a huge potential. The manufacturing of various bioproducts, biocellular and enzymic, being scale factor dependent on process biotechnologies. New and conventional processes is controlled by powerful economic and technical forces pointing towards increased levels of interdependence. The economy of scale factor is being very powerful. Engineering concerns play a great role on the technology assessment and overall success of process biotechnology. Fermentation or bioprocess technology. This is potentially a huge minefield where we have a lot of applications. So, anything concerning this has a huge role, and the safety regulations are also equally important here. So, when you look into the challenges faced by the society, one is in the bioprocess field especially, one is the availability of the raw material resources and chemical feedstocks. The second is non-conventional energy resources. Third is biological scale up and redesigning to industrial production and problems. Fourth is the healthcare problem. Fifth is the environmental concern. And sixth is the computer aided automation in biosystems. So, all these challenges do have potential answers. Biomass can be used instead of fossil fuels. So, this can become the major source from chemical feedstock. For this biomass to be utilized, we require efficient conversion of the cellulose and hemicellulose into alcohol. Okay? So, for this you require better enzymes, we require microbes whose metabolic pathways are modified to produce the better enzymes. So, how do we achieve this is first question, then what are the constraints to achieve this is the second question. So, to overcome many of these constraints, we have to select a process that would simultaneously hydrolyze both cellulose and hemicellulose to sugars. These sugars have to be converted enzymatically into xylose and glucose by yeast or bacteria, then into alcohol. Now, developing countries, there is a difference between how the developed and the developing countries utilize the biotechnological applications. In developing countries, biotechnology is very exciting and the industries find it to be a big business. And for this business to succeed, you need research and development in the areas of fermentation products, fermented food, biomass production, development of bioreactors, wastewater treatment, biomethanation, the environmental processes, along with developing academic and research institutions. So, industrial development and academic and research development go side by side in a developing country. Computer aided bioprocessing control in developing bioprocess technology has now become inevitable. Computers will have a great effect in process biotechnology with regard to automation, particularly of processes supporting genetic application. When we talk about genetic manipulation or genetic application, many of the manipulated microbes have to be controlled, they have to be contained, okay? And the scale up of this now manually becomes difficult. In such a scenario, process optimization aided by computers would have a great application, because once you feed in the data, all the processes will become automated and it will support, it will tell you where things are going wrong. So, computer aided bioprocessing is a major thrust area where a lot of potentialities can be developed, especially in food field. The next aspect is feeding the human people. In the current scenario of population explosion, as well as where we have the depletion of ground areas, crops can be cultivated, it is a challenge to provide 3 meals, wholesome meals to population. So, to meet the demand of food, feedstock and energy of the world population, this is a huge challenge. So, the use of biomass or renewable resources represents a possible long-term resolution to the problem of the dwindling petroleum reserve. The lignocellulose materials are the most abundant renewable resource produced by photosynthetics on the earth. Thus, in agricultural countries, crop production forms an integral part in process biotechnology in terms of renewable resource supply. Tissue culture. This is an application for the development of medicinal and economic plants. Tissue culture crops, for example, we have so many banana varieties or so many crop plant varieties which are clonally propagated using tissue culture. The farmers get these and plant these. So, this becomes easy against the conventional breeding technology. Fermentation technology and enzyme engineering can be used for chemicals, biochemicals, antibiotic development and other medicinal product developments through process technology, including agriculture and forest residues and industrial house waste utilization. Then we have the emerging areas like genetic engineering and molecular biology, whose applications can be applied to all these fields. Let us see the biotechnological aspects to food processing. Here, we make use of the microbial inoculants to enhance the properties like the taste, the aroma, the shelf life, the texture and the nutritional value of the food. So, we improve the microbe using genetic manipulation and utilize this microbe to improve the taste, the smell, extend the shelf life, modify the texture and the nutritional component of the food utilizing this recombinant organism. The process whereby microorganisms and their enzymes bring about these desirable changes in food materials can otherwise be called as fermentation. Fermentation processing also is widely applied in production of microbial cultures, enzymes, flavors, fragrances, food additives and the other high value added products. These high value products are increasingly produced in more technologically advanced developing countries for the use in food and non-food processing applications. So, not only food field which is being benefited, but we also have the applications of these high value products in the non-food processing field also. And many of these high value products are also imported by developing countries for their food processing applications. Technologies applied in the processing of food must ensure the quality and safety of the final product. Safe food is a food in which physical, chemical or the microbiological hazards are present at a level that does not present a public health risk. A range of technologies is applied at different levels and the scales of operation in food processing across the world. Conventional or low input food processing technologies include drying, fermentation, salting and various forms of cooking including roasting, frying, smoking, steaming and oven baking. The low income economies or countries employ these as predominant technologies for processing staple foods. Medium economies or the medium country level of processing technologies contain canning, oven drying, spray drying, freeze drying, freezing, pasteurization, vacuum packing, osmotic dehydration and sugar crystallization. The middle and the upper middle income countries utilize these. At the higher level of the economy, more capital intensive food processing technologies are utilized. They are the high temperature, short time pasteurization, high pressure, low temperature food processing, etc. This will be employed only in the higher level capital intensive food processing countries with higher per capita income. The functional additives and the ingredients produced using fermentation processes are generally incorporated into food processing operations which utilize these higher level technologies. The next aspect is the regulatory aspect. How do you monitor the safety of the food product? Traditional methods of food safety monitoring are the detection of the pathogenic bacteria. This is generally based on utilization of culture media. These are the techniques of choice on the low and the lower middle income economies which lack the resources, the infrastructure and the technical capacity to utilize the modern biotechnological techniques. So, when you go for the conventional diagnostics, The bacterial detection methods are time consuming, multi-step procedures. At least 2 to 3 days would be required because you isolate, you have to grow the organism, find out the characteristics, then confirm that it is this particular bacterium or the organism utilizing a biochemical test. So, this 2 to 3 days are required for initial isolation, followed by the requirement for several days of additional confirmatory tests. Biotechnology based methods can provide accurate results with relatively short time frame. The biotechnological developments have resulted in widespread availability of the low cost rapid methods of identification when compared to the significant cost and the time requirements of the conventional technologies. Lower middle and the middle income economies apply both traditional and more sophisticated methods for monitoring the microbiological quality of foods and the conformance with the international standards. So, to summarize what we saw in lecture 1 and lecture 2, we compared the earlier ancient biotechnological aspects, how the fermentation technology has been modernized using one, the identification of the correct microbes, 2, manipulating those microbes so as to produce better quality enzymes or isolate a pure strain so that which can be utilized in a better manner. 3, modify the fermentation process, get better bioprocess control so that your product's quality and quantity can be optimized. Then how genetic engineering technology can be utilized to produce better food crops, enhance nutrition. Then in food processing technologies and in food production technology and preservation, how to improve the shelf life. We saw the flavor saver tomato which could not be spoiled till 45 days, which was a major breakthrough. So, like that, genetically manipulated crops can be utilized for better nutrition as a biomass production, for vaccine production, what we call as edible vaccine. The third aspect is food safety, easier detection of foodborne pathogens based not only on the conventional traditional method which is time consuming, but utilizing the nucleic acid isolation and the diagnostic technology because of biotechnological applications, easier detection. And because when you, especially for a food industry, the presence identification of the pathogen becomes very rapid because otherwise your product would be rejected during export. So, the conformance to the international standard, that can be the quality and conformance to the international standards can be better utilized, can be better utilized in biotechnological applications, because when I say I used a PCR or an ELISA to test, we know that this methodology is foolproof, fail-proof, it gives so much percentage of it can be accepted, based, and it is easier, it is less time consuming. So, all these applications, biotechnological applications, have potential use in the food industry. We will be seeing individually each application in the next series of lectures. The questions based on this would be, for a particular food product, where will you apply biotechnology? Is it going to be for process optimization, or is it going to be for increasing the shelf life or improvement the quality? So, there we talk about one aspect is fermentation technology, process optimization, or a better bioreactor scale up, overproduction. The other aspect is enhanced flavor, enhanced nutrition. So, for each food product, both these aspects would have to be worked out. Thank you.